The goal of this biosensor study is to compare the ability of the 4 routinely used biosensor platforms to provide quality kinetic data and to provide insights regarding technical challenges and considerations in experimental designs for evaluating high-affinity antibody-antigen interactions.
This study answers key questions in the drug discovery field for the selection of biosensor platforms and assay formats that allow the most reliable results when evaluating therapeutic antibody candidates.
The main advantage of the study is that it provides a direct and comprehensive comparison of the various biosensor platforms with regard to data consistency, quality, and operational efficiency.
Video demonstration of the 4 protocols is critical as it helps new users with little hands-on experience learn how to set up experiments on these instruments.
For kinetic measurements in the BioCore T100, click File, Open New Method, and open the method file. Review the parameters in the method. In cycle types, set the contact time to 220 seconds for capture 1 over flow path 2, 110 seconds for capture 2 over flow path 3, and 55 seconds for capture 3 over flow path 4. The flow rate is 10 microliters per minute for all of the capture cycles. Select sample 1 and check sample solution as variable. Then, set the contact time to 600 seconds and the dissociation time to 2700 seconds over flow path 1, 2, 3, 4. The flow rate is set to 30 microliters per minute.